point first. So in the meantime, you all can chill. So maybe around 5 more minutes. Okay, before we start, just judging by the webcam, how old do you guys think I am? Like, you guys can just type in the chat. 45? Bruh, that's crazy. 26, 22, okay. 50. Yeah, I think 27 is a bit too old, yeah. But it's fine. Okay. So how was your June holiday so far? Have your teachers started going through inheritance, all of you guys? Or okay. What problems do you guys have with inheritance specifically so far, based on what your teacher has told you or taught you? Natural selection, yada yada, cross diagram, pure breeding stuff. Okay. Yes. So how this lesson will be structured is that I'll go through concepts for inheritance first, so very briefly. Then afterwards we'll do questions on inheritance law. So I'll just go through it super briefly for like those who haven't learned. And yeah, okay, 64 people. Yeah, so if you guys have any questions, just type in the chat. Don't be paiseh to ask. Just feel free to ask anything you guys have while I'm going through. Then I will answer it in the Q&A section. So yeah, no pressure. Okay, so as you all understand, this is a crash course for inheritance under Mojo Study Club. So first things first, who are we? So we're basically a tuition center that is run online, fully online. So it's built for students who want real results and real support. So the whole idea behind why we wanted to start online is because we thought that it would be a lot more convenient, saves travel time, right? And at the same time, because we don't have to pay for rental, so classes can be made cheaper for you guys while we deliver the same quality of lessons. So we have 5 young tutors who each specialize in a different subject. So as you can see, the Tele channel, we have our A Math, E Math, Telegram channel, Chem and Physics, as well as for me, Biology. And right now we have a lot of subscribers, thousands of subscribers in each of these Telegram channels. Okay, so for all the tutors, we have made it to universities such as Cambridge, NUS, London School of Economics, and King's, and hopefully we will try to impart this knowledge onto you guys. So, okay, carrying on. For this crash course, I've already sent you guys a worksheet, but if you join our classes, there are a lot more resources which you can learn from. And we have a Google Drive full of resources that we were able to help you guys score your A1 now. So as you can see, these are just samples of the resources. This is specifically for Chem that we have and Math. So if you join, you'll get weekly curated worksheets and notes, cheat sheets and summaries that will let you have shortcuts when you're studying and doing your exam papers. In addition to that, because we're fully online, right? So we'll have 24/7 WhatsApp support for you guys if you guys have any questions. So yeah. So these are just testimonies from our past students. So far, based on previous year's records, 2 in 3 of our students have achieved an A1 at their O levels. So these are some texts from our students from the past who did their O levels last year. So these are just a rundown of our tutors. For me, I'm Ryan. My name is Ryan, and I do biology. So there's Henry that does physics, Julian that does E-Math, Huang does chemistry, and Deborah does English. So these are our schedules. If you're free, you can just look through this. Yeah, so another thing I mentioned about online tutoring is that because we don't have to pay, so it's a lot cheaper for us to run our tuition center. So that's why we can make it so affordable for you guys. It's just $60 for a 2-hour lesson, while at the same time we are able to deliver the same quality of teaching that other resources do, the other tuition centers do. So if you find this crash course, you can just scan this QR code and sign up for my class. So we have 78 students now. So maybe I will just start with the topic. Yeah, currently I'm teaching pure bio. So yeah, Joan, it's only for pure bio. So inheritance, right? Inheritance is mainly like, it's not a very content heavy topic. So I'll just go through the content very quickly. Because most of the, how you score marks in inheritance is mainly through MCQs and just practical application of the knowledge that you have from inheritance. So firstly, I'll go through the difference between genes versus alleles. This is a common misconception that students have. So genes versus alleles, right? We all learn from, okay, so sorry, I need to admit more people in. So as we all learn from, the previous topics, we all know that gene is a unit of inheritance. That determines a specific trait in an organism. A specific trait, right? So these traits can be things like your eye color or your height, anything that's inheritable, basically. That's what a specific trait is, right? And so you might be wondering, what's the difference between genes and alleles? So as we all know in our previous chapters, how you illustrate genes in a chromosome is through this. So this is a gene. So the distinction between genes and alleles is that an allele would be the alternative version of a gene. So what do I mean by that? So a gene is basically an umbrella term for alleles, basically. So if you look at, let's say, your homologous chromosomes. So as we all know, homologous chromosomes are chromosomes that are similar in size, length, and they have the same genes, right? And one will come from the mom, and another will come from the dad. Okay. Any questions so far? If you have any questions, just type in the chat. Okay. One will come from the mom and one will come from the dad, right? So how a gene is shown would be like this. It will just be a line on a chromosome. Okay. And what I mean by an allele is the alternative version of the gene is that so a gene could be the gene for eye color in this case. But an allele would be specifically denoting what the specific eye color is. So an allele could be the allele for example, in this case, the dad has red eyes, so allele for red eyes, whereas the mom would have the allele for blue eyes. For eyes. So in a homologous pair of chromosomes, a normal diploid zygote would have a homologous pair of chromosomes, right? And one chromosome in each homologous pair of chromosomes, one will have an allele from the mom, and one will have an allele for the dad. So although both of these are the genes for eye color, but within the gene, there's different types of alleles. So let's have a mind map here. So for example, gene for eye color, right? But within this, there could be the allele for red eyes, allele for blue eye, allele for maybe green eyes, for example. Okay, so far, any questions? Okay, so we've already gone through the definition for alleles, but one thing you have to take note for alleles, right, is that they have this characteristic where alleles of the same gene, so for example, alleles coding for the same gene. So for example, alleles for, we've already gone through eye color, right? So we will just use the eye color example. Alleles that code for example, eye color will occupy the same locus on a pair of chromosomes. So does anyone know what the definition of locus is? You guys can just type in the chat. Okay, so okay, don't be too scared by the term lah. So what a locus essentially is just a specific position of a gene on a chromosome. Okay. So just now, I don't know whether you guys noticed, but the alleles are always aligned with each other. So let me illustrate this again in another example. So we all know homologous chromosomes, right? One from the mom and one from the dad. Okay. Okay. So let's say another gene could be another trait that could be passed down is the ability to roll your tongue. So let's say the dad can roll his tongue, so he has an allele for that, right? So we'll just put this allele in blue. So blue is the ability to roll tongue. Allele for ability to roll tongue, whereas the mom has the allele for, let's say the mom cannot roll her tongue, so let's say allele that makes mom unable to roll tongue. So you can see that in a homologous pair of chromosomes, the allele that codes for the same gene, they will always be aligned with each other. So that's basically what it means. So a locus is just a fancy term for the position on a chromosome. Okay. So don't be too scared by this definition. Okay, so we've already gone through the definition, the distinction between genes versus alleles. So I'll just take this first. So I'll just flash some questions for you guys to reinforce this concept. Give me a while. So if you guys refer to the worksheet I sent you, it's basically split into learning objectives, right? So we've already gone through this learning objective, gene versus allele. So now we'll go through a question. So let me do a poll, and then you guys can answer question 1 first. Okay, so question 1, A, B, C, D. I'll launch it right now. You guys can see the poll, right? Okay, I just realized I just saw the questions on the chat. Metal Pro, I'll answer you after this question. And okay, Berlem is correct. Location of the gene. So first question, what is the relationship between a gene and an allele? So we've already gone through this just now, so it should be quite simple for you guys. So we have 13 votes so far, 14 votes. Yes. Okay, is everyone done so far? If okay, I realize that half of the people haven't voted yet, but okay, I'll give you guys 1 more minute. You can see on the tabs below. Yeah. Um. Okay, so far the majority of you guys have voted C. I see a few voting A. So if we first go through the first question, so what is the relationship between a gene and an allele? So some of you guys voted A, right? So A states that an allele codes for a particular characteristic or protein, or a gene is one of the many forms of the same allele. Okay, so it's actually terbalik lah. So the gene will be the one coding for the particular characteristic. So by characteristic, it means things like eye color or your height, right? Whole lot of other things can be your hair color as well, okay, etc. And then they say, well, a gene is one of the many forms of the same allele. So it's terbalik lah. So basically what it means that this should be gene, while this should be allele. So maybe you guys read the question wrongly. I'm not very sure, but yeah, so this is wrong. So one guy voted B. So it says an allele is a rod-like structure visible in the nucleus during cell division, while a gene is one of the many forms of the same allele. Again, this is wrong. This statement is wrong. A gene codes for a particular characteristic, yes. Or a protein, while an allele is one of the many forms of the same gene. Correct. So the answer is C. Again, I state again. So a gene is a blanket term for an allele. So for example, a gene for eye color. Within a gene for eye color, you have alleles that code for the specific eye color. So again, I reiterate, allele for blue eyes, or can be, for example, allele for red eyes, and so on and so forth. Think of all the many colors, allele for brown eyes, allele for green eyes, things like that. So question 1 is C. Okay. So we move on to question 2. Which of the following statements is not true? Let me pull up the poll for question 2 right now. Jaden, this class is for pure bio, but combined bio have some content that is tested that I'll be going through also. Vanessa, what is sister chromatids? Okay, I'll take note of Metal Pro and Vanessa Lau's question. Homozygous. Okay, I will go through that later as well. So for Metal Pro and Ryan Tan, I will be going through your questions because it's part of the syllabus as well. Vanessa, I'll go through it later as well. So don't worry. Okay, so homozygous just means that so within a homologous chromosome, right, you have one allele from the mom and one allele from the dad. So again, I'm going to draw this. One allele from the mom and one allele from the dad. So red is mom, blue is dad. So homozygous means that the so it just means that the alleles from both the mom and dad are the same. So in this case, if you want to label this as homozygous, if you want to label this as homozygous, this should be, for example, allele for blue eyes. And this should be allele for blue eyes as well. So that's when the alleles are homozygous. So I'll go through this question now. If everyone's voted. Yeah, I'll give you guys 30 more seconds because we have to rush through this thing. Okay. So there's 7 votes. I'll just end the poll now. So we have 7 votes for A, 23 votes for B, and 6 votes for C. So it shows that slightly harder for this question. So they say which of the following statements is not true. Number A is that alleles of the same gene can be found in the same gene loci of homologous chromosomes. So this is correct, right? Because just now I went through that. Just now I went through that alleles will always have the same loci, right? So if the alleles that code for the same alleles that code for eye color, for example, so it doesn't matter whether it's allele coding for blue eyes or allele coding for red eyes or allele coding for green eyes, they will always be located on the same loci. So loci means position, right? So they will always be aligned with each other like this. Okay, so first one is obviously correct. So we rule this out. Alleles of the same gene loci must be homozygous. That's not true. So, for example, parents who have different eye colors. So, for example, a mom has the allele for blue eyes, whereas the dad has the allele for red eyes. The son will have the alleles for blue eyes and red eyes. So alleles from both mom and dad can either be the same or they can be different as well. So this is not true. So the answer is B, but I'll go through C. Homologous chromosomes have the same sequence of gene loci. Okay, this is not true as well. I mean, this is correct, sorry. Sister chromatids have the same sequence of gene loci. That's correct as well. So the answer is B. Okay, so we'll go through. Okay, another thing I have to go through with you guys is this. Okay, so just go down to a new page. So this is not actually inheritance, but I'll go through with you guys. So a lot of students also have this misconception between genes versus chromosomes versus DNA. So we learned already that in this chapter, genes are just a unit of inheritance that determines a specific trait in an organism. Okay, but in the previous chapters, we also learned that gene is a short sequence of DNA. So whereas chromosomes are just DNA that's been coiled up and compressed together. And the DNA is just the molecule itself, right? So visual representation. You guys have probably seen this diagram before. So you can see in this diagram, right? DNA will just coil up to form chromosomes. Form chromosomes. Like this. So you can see like the DNA molecule that's been coiled up inside a chromosome over here. So whereas the gene will be DNA that's been coiled up in a chromosome. So if you see here, right, this will be the DNA, and a gene will just be a part of the DNA. So for example, can use this example again, eye color. So this will be the gene for eye color, and it's just going to be a short sequence inside the DNA. So the DNA, realistically speaking, is super long now. So it'll be like this, right? Sorry. And within this DNA, there will be many types of genes, thousands of genes that will be inside one long DNA molecule. So for example, this is a gene for eye color. This could be the gene for hair color. This is not an accurate drawing, just for you guys to know the concept. And this could be the gene for, let's say, ability rotang. Okay, so when DNA has been compressed into chromosomes, that's when you show a gene like this. So, for example, this could be a gene for eye color again in red. Then this could be the gene for hair color in black. So along the length of the chromosome, there will be many different types of genes, basically. and this could be a gene for ability to grow tongue. So this is the visual representation of a gene inside the DNA. Okay, over here, circled in green, visual representation for the gene inside the DNA, whereas circled in purple is the visual representation of a gene inside the chromosome. Okay, so I'll just flash my screen, and then we will go through another question. So yeah, important to take note lah, genes versus chromosomes versus DNA, because a lot of students mix up the concepts between these three. Okay, so I'll just continue presenting question 3. So you guys just do this question first. What is the set answer if they ask you what's the relationship between the DNA, genes, and chromosomes? Oh, okay. I'll go through it after the lesson during Q&A. So I'll just write down a list of questions that you guys have asked. So I'll go through in Q&A. Are all chromosomes in the body identical, or are they all different? Are all chromosomes in the body identical, or are they all different from different chromosomes have different codes to gene? Different gene to code for different stuff. Okay, so not all chromosomes in the body are identical. So remember, within a human body, within a human cell, we have 22 pairs of homologous chromosomes and 1 pair of sex chromosomes. So within this 22 pairs of homologous chromosomes, I don't know whether you've seen the karyotype, but they are all of different lengths. And each pair of homologous chromosome will have genes that code for different traits. So no, not all chromosomes in the body are identical. They are all of different length, and they all have different genes that code for different things. So this topic also goes through the concept of karyotypes. So karyotype is basically just a diagram that shows all the chromosomes that are in the human body, 46 chromosomes. 2 out of 46 of them are sex chromosomes, and the other 44 are homologous chromosomes, are pairs of homologous chromosomes. Yeah, autosomes are just chromosomes that are not sex chromosomes. So 44 autosomes, if you want to put it more specifically, and 2 sex chromosomes. Okay. So A, most of you have voted. Chromosome consists of many genes arranged in a particular sequence. So yeah, I think most of you have gotten this concept, right? One voted for B. A gene is an alternative form of the chromosome. That's not true. So a gene is located within a chromosome, but they're not alternative form of the chromosome. A gene codes for a protein known as a chromosome. Chromosome isn't a protein. So the first part is true. A gene codes for a specific protein. So remember, you learn in transcription and translation, the mRNA will copy the sequence on the gene, right? And then it will get out of the nucleus. And from there, it will undergo translation to form the code for a specific protein. But no, a gene doesn't code for the chromosome. A gene is located in the chromosome. Always remember that. And no one voted for D, which is good. Okay, so I'll end the poll now. Okay, so all okay so far. Any other questions? So now I'll go through. Okay, so based on our checklist, we have already gone through genes versus alleles, right? And the relationship between genes, chromosomes, and DNA. Remind me later to go through the set answer for this question. So now we have dominant, recessive, and codominant. Okay, so okay, so we already learned what alleles are. They code for a specific trait. They code for a specific trait. So, for example, allele for black hair, allele for brown hair, for example, right? So alleles can be further categorized into dominant and recessive. Okay. So what do I mean by that? So remember I said in the human body, an individual will have one allele from mom, another one from dad. Okay. So how these alleles are shown, right? It's basically in letters. So again, maybe we use the rolling tongue example because it's convenient, right? So let's say this guy has a mom that can roll a tongue. But then in this case, this guy has a dad that, okay, let's call him Zachary, for example. So Zachary has a mom that is able to roll a tongue. Whereas the dad has the allele that does not allow him to roll his tongue, so he cannot roll his tongue. So the alleles for that Zachary will have will be one that can roll tongue, one cannot, right? So how this is shown, right, is like this. Okay, another thing you guys have to take note that rolling tongue is a dominant allele, whereas cannot roll tongue is a recessive allele. So dominant alleles are usually shown in capital letters, whereas recessive alleles are usually shown in small letters. So Zachary, for example, will have capital letter R. Okay. Whereas the dad, okay, from the mom side, Zachary will have capital letter R. Whereas from the dad side, Zachary will have a small letter R. So in this case, the small letter R is the recessive allele that's shown, which is cannot roll tongue, right? And the big letter R will be the dominant allele that's shown, which comes from the mom, can roll tongue. So dominant alleles and recessive alleles are denoted like this. So recessive allele will always be in small letter, while dominant allele will always be in big letter. So in this case, Zachary's alleles will be shown like this. One will be big R, and one will be small letter r. Okay. So by definition, dominant just means that is the allele to always be expressed regardless of whether the individual is homozygous, which really went through, right? Homozygous just means that they will both have the same allele. So for example, 2 capital letter R's or 2 small letter R's, okay, or heterozygous. Whereas recessive allele are just alleles that will always be expressed over the dominant, yeah. So in this case, Zachary has 1 dominant allele, right, and 1 recessive allele. So which trait will be shown? Will Zachary be able to roll his tongue, or can he has 1 dominant and 1 recessive? So someone maybe type in the chat. Yep, can roll, right? Because again, the definition of dominant allele is that it will always be expressed regardless of whether the individual is homozygous or heterozygous. So in this case, the individual is obviously heterozygous because he has different alleles, right? One from the mom is able to roll tongue, the one from the dad, the allele shows that cannot roll tongue. So in this case, because the dominant is always expressed over the recessive, so big R will always go over small r, will always be expressed over small r. So in this case, Zachary can roll his tongue. So these are very simple concepts. Again, inheritance is quite simple. It's just more about being able to apply what you know. So inheritance involves a lot of practice questions, basically. Okay. So again, let's show Zachary's example, right? 1 big letter R and 1 small letter r. So in this case, is the person homozygous or heterozygous? Zachary. Is Zachary homozygous or heterozygous for this trait? Yep, hetero, right? Because again, hetero is just the opposite of homozygous. Really went through that homozygous means that the alleles are the same. So heterozygous just means that 2 different alleles. Jaden, what's homozygous and heterozygous? Okay. Homozygous just means that, okay, I'll go through again. Homozygous just means that the 2 alleles for the particular gene are identical. Okay. Yeah, for Jaden, the 2 alleles for the particular gene are identical. So again, 1 from the mom, 1 from the dad. Mom, dad. And let's say they both brown eye color. Allele for brown eyes. They both have the allele for brown eyes. So the dad will have the allele for brown eyes. And the mom will have the allele for brown eyes also, right? So this means that Zachary would be homozygous for eye color because the 2 alleles are identical. What? The mom and dad gave him both the alleles for brown eyes. So that's what homozygous means. Then heterozygous is just the opposite. So Jaden, does that clarify your question? Okay, I'll just assume it did. Okay, nice. Okay, so we've already gone through dominant, recessive. So there's a caveat to this, there's an exception to this. So there's this thing called codominant. Okay, so what codominance basically means is that both alleles will have an equal effect on offspring's phenotype. So what do I mean by that? Okay, so codominance only applies to specific kinds of traits. So, sorry, I need to admit someone else wants to join the call. Sorry, give me a while. Okay, nice. So again, codominance only applies to specific kinds of traits. So codominance happens in things like your blood type, for example. So I don't know whether you know, but there's this blood type called AB. You guys probably have already learned it, right? So codominance only applies to certain kinds of traits. So codominance are things like, for example, your blood type, or another popular example would be the color of the wool of sheep. Okay, so I will just use blood type as an example to highlight codominance. So your blood type, within your blood type, there are 4 blood types in the O-level syllabus, right? So there's blood type O, A, B, and AB. So the alleles for this are IO, which is IA and IB. So we all know that IO, the allele for blood type O, is recessive. Okay. Whereas the allele for blood type A is dominant. And the allele for blood type B is dominant. Okay, so if a person has blood type O, right, it's quite clear cut. The possible combinations for alleles that the person will have. So possible allele combinations would be IA, IO, IA, sorry, this is wrong, sorry, my bad. It would just be IO and IO. Because remember that IO is recessive. So this is the only combination. It cannot be IO because then I would express itself over O, right? So blood type A, possible allele combinations would be IA, or IO, because remember that IA is dominant. So it will always express itself over IO. So that's the reason why IA and IO will give you the blood type A. Then you have blood type B as well. So blood type B will be IB, or IB, IO. So we go to the last part, blood type AB. Okay. So this is when codominance comes into play. Blood type AB will be IA and IB, like this. So remember, we see codominance is when both alleles have an equal effect on the offspring's phenotype. So you can obviously see it here, right? I and IB, they're codominant. So 50%, 50%. And they'll form this new blood type called AB. If the crash course has been helpful so far, spam some emojis right now. Okay. Thanks. Anyways, blood type AB, right? So that's what codominance basically is. So you guys might be seeing that in the definition for codominance, there's this thing called phenotype. So Another thing you guys have to take note is the distinction between phenotype and genotype, because some students also confuse between the two. Okay, so phenotype versus genotype, right? Okay, so genotype is basically what we've already gone through. So a genotype is just basically a combination of alleles coding for a specific trait that code for the same trait, basically. So we've already been drawing genotypes, actually. If you guys haven't noticed, combination of alleles that code for a specific trait. So things like what I've already drawn, for example, Zachary's genotype, right? Specifically for his ability to roll his tongue would be one big R and one small letter R. Okay, so that's just an example of what genotype is. It's just on face value, it's just the two letters lah. Okay, so a genotype could be, for example, RR, or could be two big letter R as well, or for any trait. So for in this case, I'm just showing the genotype, the possible genotypes for ability to roll tongue, right? So there's these three possible genotypes. Okay. So we go to phenotype. Okay, so phenotype is just what you derive from the genotype. So it's just a trait you get from the genotype. So we can look at blood type again, right? So in this case, right, I've already gone through blood type, so I'll just use it to be convenient. So in this case, the phenotype would be the type of blood. So blood type O, A, blood type B, and so on and so forth. Whereas the genotype, in this case, would just be a combination of alleles, right? Again, I reiterate, the combination of alleles that will code for the blood type in this case. So the genotype in this case will be IO, IO, IA, IO, or IA, IA, or IA, sorry, IBIO, or IB, IB. Then the last one would be blood type AB, right? Codominance. Because A and B will both express themselves equally. So IA and IB. Okay, so far? Any questions? Okay, so just remember that you always derive the genotype. You always derive the phenotype from the genotype. So the genotype will be what gives rise to the phenotype. Okay. Any questions so far? No questions. Okay, so back to our checklist. We have done all this so far. So we have done genes, chromosomes, and DNA relationship, genes versus alleles. We have already gone through dominant, recessive, and codominant, genotype, phenotype, homozygous versus heterozygous. We've already gone through as well. They're all very easy. So I'll just flash some questions now because I stress that inheritance is really just about practicing because the concepts behind them are very simple. So let's do question 4. Okay, let me launch a poll first. So question 4. Okay, so I see that most of you guys have voted C, whereas some of you guys have voted B. Okay, so I'll show the results now. Okay, so this is actually a trick question. So just now I went through that. I did say that yes, phenotype is determined by genotype. We've already established this. So these two are correct, right? So the last one, B, okay, environmental factors. So I didn't go through that. So yes, you get your phenotype from your genotype mainly. So for example, blood type, right? I've already gone through. Genotype is just a combination of alleles that code for the trait. So in this case, you've already seen that, yes, you do get your genotype from your phenotype. You do get your phenotype from your genotype. But another thing you guys have to take note is that for phenotypes specifically, they are also influenced by environmental factors. So this is one thing that you guys have to take note. It's quite important. So I'll just give you guys a list of possible phenotypes. So things like your height, things like your skin color. Okay. They're also influenced by environmental factors. So for example, your height. What could possibly affect your height? Things like lack of nutrition, right? These are factors that are environmental that could affect your phenotype. So for example, skin color as well. Even though it's genetically determined, okay, how dark your skin is genetically determined at birth, which is obviously uh determined by your genotype. Another thing you guys have to take note is that skin color could also be influenced by things like sun exposure. Right? So it's not entirely just based on your genes alone, although your genes do play a part. Things like uh your exposure to sun, for example, or whether you do skincare or whether you put on sunscreen also can affect your phenotype as well. In this case, skin color. So just take note of that for question 4. We'll go back to question 5. Sorry. Yes, question 5. I'll flash it right now. So if allele T is the dominant allele for cows with straight tails, while the allele small letter t is a recessive allele for cows with curly tails, what genotype will a purebred cow with straight tails have? Take note of the term purebred. Just means homozygous alleles. So this is pretty easy as well, quite clear cut. So this question is just testing on whether you know the meaning of purebred. Okay, I'll give you guys 20 more seconds. So for phenotype, is it always determined during birth? Or can it change within your life? Okay. Sorry, I'll go through a question after this. After question 9 and 10. So question 5. Most of you guys voted A. 29 votes for A and 3 votes for B. So I think you guys do know the definition. For B, for those who voted B, just remember that purebred always means homozygous. So in this case, the genotype is heterozygous, right? So obviously this is wrong. This is correct. So A is correct. So question 9. Okay, question 9. So pretty split for question 9. So some of you guys voted for B, a few voted for A, but most voted for C. So okay, for question 9, which of the following cannot be observed in the ABO blood group of humans? Cannot be observed. Codominance, as I already said, you can see in blood type AB, right? When the alleles are AI and AB. Co-dominance happens. They both express themselves equally, and this is what causes blood type AB to happen. Complete dominance. So, some of you guys would look B. Complete dominance just means that one is completely dominant over the other. So, you can see this in your blood type A and B. So, possible allele combination for blood type A and B could be IA and IO, or IB and IO. So this is for B, this is for A. Sorry, this is for A, and this is for B, right? So no, complete dominance does happen in blood types, in the ABO blood group of humans. Because remember that IO is recessive, and IA and IB are dominant, right? In this case, IA will express itself over IO to give you blood type A, whereas IB will express itself over the recessive IO to give you blood type B. So complete dominance does happen. So continuous variation is correct. So brief recap on continuous versus discrete. So I haven't gone through this yet. So for traits, they can either be for traits in humans, right? In humans, right, they can either be split into these two categories. So discrete and continuous. So discrete traits are things like your for example, your blood type or your ability to wear a tongue, or whether you have attached or detached earlobes, or for example, eye color. Whereas continuous can be things like your skin color or your anything, your height, example. So looking at this, can anyone tell the difference between discrete and continuous? Anyone? Discrete versus continuous traits. Just type in the chat if you're comfortable with giving an answer. Discrete versus continuous difference. Anyone? Just looking at these traits, both of these traits. So one of you guys voted no effect on environment, right, for discrete. Okay, so yes. So discrete traits are, for example, and continuous. One of you guys both said that continuous has a lot of variation. So let me pull out this diagram. Okay, maybe I'll draw it myself. Anyways, discrete traits are usually shown like this. So for example, this is the graph for eye color. Okay, it'll be shown like this: blue, red, green, brown, maybe. Whereas continuous traits are usually shown like this. So there'll be a normal distribution for continuous traits because it's over a range, right? So continuous traits will always be shown over a range, whereas discrete traits are a few distinct phenotypes. So let's say, for example, this is 25% blue, and then 25% green, and 25% red, and 25% brown, and so on and so forth. But continuous traits are always shown over a range. So basically, think of continuous traits as traits that are measurable, basically. Things that you can attribute numbers to it. So things like your height, right? So let's say the average person's height of a guy in Singapore is like 1.7. So most people will fall within this 1.7 range. And then let's say this is like 1.8, and then this is like 1.6. So continuous traits, always think of it as things that are measurable. Another thing that you guys said also is that discrete traits are usually not affected by the environment. So this is true. Can your blood type be affected by environment? No. You're born with your blood type. Can your ability to roll tongue be affected by environment? No. You're either born with the ability to roll tongue or not. You cannot train that. You cannot change the fact that you have attached or detached earlobes. You're just born with it as well, right? Your eye color as well. You can't change that. You see things like your skin color. Obviously, you can change your skin color. Just sun exposure, right? As we said just now. Affected by environment. Things like your height as well. Affected by environment. Nutrition, for example, plays a big part in how tall you eventually end up being. So yeah, the main two things you have to take note for distinct versus continuous is that, discrete versus continuous is that number one, discrete traits, few or distinct phenotypes. So distinct as in, you can obviously tell the difference between a person who has blood type A, B, or AB, blood type A, B, or AB, or O. And you can tell the difference between someone who has attached or detached earlobes. It's very easy to tell. And there's only a few. So for blood type, for example, there's only 4 types, right? Attached or detached earlobes. There's only 2 types, right? You can either only have attached or detached earlobes. For example, your ability to roll your tongue, you can either only roll your tongue or you can't. So there's only a few phenotypes. Whereas continuous traits is over a range, right? There's such a spectrum over what height you can be. It can be 161, it can be 162, it can be 163, it can be 171. It's always over a range. Okay. So another thing you guys said, so it's correct. Discrete traits can be usually not affected by environment also, as I pointed out just now. Okay. So just remember this diagram. It will help a lot. So let's put out another example, right? Continuous traits diagram for skin color, for example. So from light skin to dark skin. Most people will probably fall between very dark and very bright, right? Very light. So yeah. Okay, so I'll flash another question relating to this, and we'll see whether you guys understand or not. So if you look at our checklist, we're already done with all these, right? Continuous versus discontinuous traits. So I forgot to say that discrete is also known as discontinuous. So they're both the same thing. They're interchangeable. Okay, so we're already done with our whole checklist. So I'll just flash. Okay, if given a question, they show like a. If given a question where they show like a graph and then ask you, is this discontinuous or continuous variation? How do you answer this? Set answer. Okay, so Saga, you asked set answer for relationship between DNA, genes, and chromosomes, as well as this. I'll go through this after the MCQs lah. Because MCQs are quite foundational, so I just want you guys to know the concept first before we go into the structured questions. Okay, is that fine? Is that okay? Yeah. Okay. So back to what we have learned so far. Okay, this is the karyotype that I was talking about lah. The diagram that shows all the chromosomes, right? So one of you guys asked, are the chromosomes all the same length? You can obviously see that they are not. So you can see that 22, for example, is shorter than 1 because they have different genes that code for different things. Each pair of homologous chromosomes have different genes that code for different things. So mom, dad, so forth, so on and so forth, right? And they are obviously of different lengths. But within a pair of homologous chromosomes, the chromosomes are obviously the same length because you have the same genes, right? You have the same genes that are located on the same locus, position. Okay, so for example, this could be red eyes, and this could be blue eyes, and they're always located next to each other. Always remember that. Alleles always have the same loci, always align along the same loci. Okay, so question. Okay, let's do let's carry on from let's do question 10. Brendan has blood group AB while Trina has blood group B. Which of the following phenotypes is not possible for a child to both of them? Okay, I'll launch the question. Just vote now. Yeah, I think this is quite a simple question. I think I might just skip it. But yeah, most of you got it correct. Actually, all of you got it correct. So D, obviously, because this will give you blood type O. This will give you blood type A, right? This will give you blood type B. And oh, shit, sorry. I didn't read the question properly, my bad. Not possible for a child born to both of them. So in this case, you learn this thing called monohybrid cross. So monohybrid cross, how it works is that you have one parent that is AB and the other one is B, right? So we'll list out the possible combinations of alleles for AB, which is just IA and IB. And B has two possible combinations, right? So you have either IB, IO, because obviously B is recessive over O, dominant over O, right? So it can be IB, IO, and it can be what's it called? IB and IB. So you learn this thing called a monohybrid cross. So monohybrid cross, basically what it means is just it's just a diagram that will show you the genotype for the offspring. So from AB, you have Brendan, right? This is Brendan. And then you have Trina. So how monohybrid cross works is that you just do this. It's quite simple. Just do this. So from Brendan's side, you have IA, IB. And then from Trina's side, you have IB, IB. So we just do this. And the possible genotypes for offsprings will be located here. Maybe. Then for the second combination, second possibility, so we really have possibility A, right, of them mating. So we go on to possibility B. So this A. Possibly B is just like this, lor. You just cross them together again. Remember this thing is called monohybrid cross. So it's what you do when you want to know the genotypes of the offsprings, lah, basically. And IBIO. So in this case, you can see that IIB is obviously a genotype that can be shown. That an offspring could possibly have IBIB as well. Because you see over here, IBIB is one. And IIO as well, because see here. So yeah, obviously the answer is B, which most of you got correct. So any questions on monohybrid cross? If not, I'll just go to the next question. Just raise up your hands if you want me to move on. Okay. Question 12. In the human ABO blood groups, how many genotypes are there for each of the expressed phenotype? So yeah, this is good. Most of you got a concept already. So yeah, question 12 is question 12 is D. So we have one guy that voted for A, one guy that voted for C. So remember that A has two combinations as shown here, because A is always like dominant over O, right? So there'll be another combination where IA and IO will give you blood type A, and for B as well, because they're both dominant over O. Question 13. Okay, yeah, that's great. Yes, nice. So great, getting the hang of it. Child's blood group A, which so yeah, obviously you can cross this again, right? And see that say the father's blood groups IAIO or IAIA. You have IA from the mom. So you can obviously see that the child will be blood type A. So, correct. Don't think I have to go through this in detail. Okay, so using genetic diagrams to solve problems involving monohybrid inheritance. We've already gone through that. Test cross. Okay, this is when it gets a bit tricky. Have your teachers gone through test cross with you? Or are you guys very familiar with this? Using a test cross to determine the genotype of individual dominant trait? No. Okay. Okay, so test cross. So this test cross, right, uses a monohybrid cross. So the monohybrid cross is what we introduced now. It's just the two alleles from the mom and one allele. We have the two alleles from the mom, then two alleles from the dad. Then you just cross it to see the genotype of the offspring, right? So 25%. So in this test cross, in this monohybrid cross, you can see that 50% chance of offspring to have blood type A and 50% chance for offspring to have blood type AB. Okay, so we have already gone through this. This is very clear cut, very easy. So a test cross is used to determine the genotype of an individual. Okay, so it's only used to determine if an individual has an unknown genotype. You don't know the genotype of his offspring. Okay, or of the individual. Because and this only happens when the individual has a dominant phenotype. So why is this test cross used? Because in a dominant phenotype, right? So for example, what's a dominant trait? Ability to roll tongue. A dominant trait will be ability to roll tongue. Let's say Zachary has ability to roll tongue. But you're not gonna be sure of what genotype he is. What you only know his phenotype, which is that he can roll his tongue, right? But you're not sure whether his genotype is either R, big letter R, small letter R, or two big letter R's, because you just won't know his genotype. Okay, so what a test cross does is that you see that Zachary has a dominant phenotype, right? He can roll his tongue, but you wouldn't know his genotype, right? Just by looking at him. So what you do is that you use a monohybrid cross. So you do an experiment whereby Zachary breeds with another individual. So he's crossed with individual that is homozygous recessive. So what you always do is you cross with an individual that is homozygous recessive. So in this case, Zachary will be the individual with an unknown genotype, right? But you know that he has a dominant phenotype. And you cross it with an individual that is homozygous recessive. So in this case, Zachary could either be RR or R, R. And you cross with this individual that has two small letter R's. Okay. So the whole purpose, right, is to find out the genotype of Zachary. So how you do it is that you look at the offspring that Zachary has with this individual. So from there you can determine. Okay, so what do I mean by that? So if you look at, let's assume that Zachary has two big letter R's. So this is Zachary, Zachary is the one on top. Let's say Zachary has two big letter R's, and then he's crossed with this individual with two homozygous, there's homozygous recessive, so two small letter R's, right? So the offspring will be like this. But let's assume that Zachary has one big letter R and one small letter R. And then you cross like that. You can see that the offsprings will be different. So assuming that Zachary has we already know that he has a dominant phenotype. We know that he has a dominant phenotype, but assuming that he has a genotype that is the first one here, which is two big letter R's, okay, what will happen that will be that all his offspring can rotang, because they will all have the genotype of big letter R and one small letter R. But assuming that Zachary has one big letter R and one small letter R, 75% of his offspring will be able to rotang, but 25% cannot. So it's two small letter R's. So basically what this test cross does, right, is that you will always cross with an individual that is homozygous recessive if you don't know the genotype of Zachary, but you know that he has a dominant phenotype. So when you look at the offspring, based on the results of the offspring, you can see that you can infer what Zach's genotype is. So a question could be like this. For example, Zachary, unknown genotype. But cross with homozygous recessive, but that's cross with homozygous recessive individual. What will happen is that then, for example, the question could ask offspring of Zachary and this individual. 75% can rotang, 95% cannot. So right off the bat, you look at the offspring, right? 75% can rotang, 25% cannot. So although you don't know Zachary's unknown genotype, but you can see the results of the offspring. And then from there, you can infer that Zachary will have a genotype of one big letter R and one small letter R. So you guys understand the concept behind test cross now? Yep. Okay. So yeah, test cross is a concept that will be tested as well. So just remember, you always, if you don't know the genotype of the person, but he's showing a dominant phenotype, then you just always cross with the individual that is homozygous recessive. Then from the results of the offspring, if it's 75% dominant, 25% showing a recessive phenotype, then Zachary will have, that means that Zachary or the unknown genotype individual will have one big letter R and one small letter R. Then if Zachary has a dominant phenotype and the genotype is two big letters as well, or two dominant alleles, it means that all offspring will have the dominant phenotype also. So yeah, so that's the concept behind test cross. One more thing that you guys can take note for you guys to cheat the system so that you guys don't take too much time is using this thing over here. So if you guys are able to practice enough and you familiarize yourself with the results of the offspring from monohybrid crosses, then it will be very easy for you guys to do your family tree diagrams, to do the family tree questions. So you guys can just memorize results of offspring from crops. So if you guys know the results instantly, then you guys will be able to skip the process of doing a monohybrid cross. So you memorize the results of offspring from the cross in order to infer what the parents' genotypes are. So it'll be very easy for you guys. So from today's worksheet, make sure, I think this is quite important. Just being able to remember this whole diagram will save you a lot of time if you're doing things like your family tree questions, which are very time consuming. Okay, so enough with that. Let's do question 6. Sorry, I forgot I didn't put a poll. My bad. Question 6. Sorry guys, my bad. Launch. So this is related to test cross, right? Because you have this allele, you have this fruit fly that is, you don't know the genotype, so it's an unknown genotype. But you know there's either or RR because it's red eye. But then brown eye, you already know it, because for an individual to have a recessive phenotype, always remember for an individual, recessive phenotype, it has to be homozygous. Okay. It has to be homozygous because RR will give you red one, but RR will give you brown. So it always has to be homozygous if you're showing the recessive phenotype. So you already know the genotype for the brown-eyed fly. You already know the genotype is RR, two small letter R's. Okay. So this is a test cross, essentially. So they said a total of 120 of the offspring had red eyes, and the rest had brown eyes. Okay. Okay, one question. You can use a Punnett square, but then again, it's time consuming lah. So it would be preferable if you just know the results straight away. So remember what I showed you guys, this thing over here. It'd be preferable if you just memorize the results, and it'll be very easy for you guys. So let me show an example. So okay, most of you guys voted most of you guys voted B, which is okay. B, let me do a question now, sorry. Okay, so 6, right? So you have you have 100. You have this guy, red-eyed fly. So again, you don't know the genotype, right? And then you have the brown-eyed fly, which obviously you know the genotype because I reiterate that for an individual to show a recessive phenotype, it always has to be homozygous. Okay, homozygous for the recessive alleles. Okay, so the question said that let's go back to it. The question said that a total of 120 of the offspring had red eyes and the rest had brown eyes. So if you go back to my test cross, right? If you go back to this, right? Remember that if in a test cross, right? If this guy is like that, if the red-eyed fly is homozygous dominant, which means that he has two big letter R's, all the offspring will be red-eyed fly, what? Because the genotype of all offsprings will 100% be the genotype for the offsprings will 100% be one big letter R and one small letter R. And because big letter R is always dominant over small letter R, remember? So 100% of them will be red-eyed. Okay. So this links to my test cross, right? Whereas if the brown-eyed fly, if the red-eyed fly is just like this, one big letter R, one small letter R, and you cross it like this, it will be 50% brown. Offspring will be 50% brown because homozygous. And the other 50% will be red-eyed because that. So yeah. Okay, sorry. Oh, my bad. I did this question wrong. I did this example wrongly. Should be 50% here also. Yeah. So what you do is just you look at the offspring, right? They said that the rest have brown eyes. And then you already automatically know that the other one will be 50%. So it helps you save time because you already know the results. You know that it's 50%, 50%. If you're crossing a heterozygous dominant, it will always be 50%, 50%. If this guy is big letter R, one small letter R. So answer is B in this case. Question 7. Here, let me do something. Sorry. Yeah. So what you learn from the test cross, right, is that what you learn from the test cross. The moral of the story is that um, if unknown individual for example, the red-eyed fly, right, in the previous question is homozygous dominant. So for example, like that, all offspring will be red-eyed, will be like, but if the unknown individual is 50% will be brown, 50% red. Okay. So you can see the results here, right? If it's homozygous dominant, 100% will be red-eyed. If it's heterozygous dominant, which is this, then 50% will be red and 50% will be brown. So you guys can just use this concept to answer the questions fast lah. For question 6 specifically. Okay, sorry, back to question 7. Okay, for question 7, most of you guys voted D. In the sheep, LOW with coats of white woods coming over LOW. It's quite easy also. Yeah, another pro tip is that if they both have the same letters, right? So if the capital and the small letters are the same, what you usually do is that in a mono-hybrid cross, you don't confuse yourself. So this is big. The one on top of two big W's, right? You just put a apostrophe here. Do not confuse yourself. So this will be the small letters in apostrophe, circled in blue. Okay. Then you just that. So yeah, 0% will be black wool. Okay, I don't really think we have much time to go through the rest of the questions. I'll probably flash through one more question, then we'll call it a day. I haven't gone through actually. I haven't gone through I haven't gone through mutations yet, and yeah, I haven't gone through mutations yet. But I'll just flash this question 30 and 31. Then we'll call it a day. Yep, Javier. Okay. Okay, so most of you guys have voted B. Yeah. So one guy voted C and one guy voted D. So this is quite simple. So heterozygous pair, which means that different letters, right? With erect ears is crossed with homozygous pair. Erect ears is the dominant one, so let's just label it as E crossed with homozygous pair with droopy ears, which is that. So just do this. Wait, sorry. So 50%, 50%. So maybe quite easy. This is question 30. Ignore this is question 30. So they said two erect eared offspring of E. Let me start another poll. 31, sorry. Yep. So this will be the last question. If you guys have any questions, you can stay back after class, after this crash course, and I can entertain them. So so they said mention, right? So the erect ear offspring will be the heterozygous offspring. So they will have a genotype of one big letter E and one small letter E. Yeah, I can give you guys an answer sheet. Sure. Okay. I'll just go through this very quickly. So, which of these is the most likely reason for the observation? So some of you guys voted A. So A is wrong because okay, it really depends on how they ask the question. So if this was 1,000 piglets instead of 10 piglets, then A, B, or C could very well be the answer. But because they said it's 10 piglets, right? So that's why the sample size of 10 offspring is statistically too small to be accurate. So one more thing you guys have to take note is this concept of sorry, there's so many expected versus observed ratios. Okay. So expected ratios is what you get theoretically. So for example, in the previous question just now, theoretically, the expected ratio would be, where is it? Sorry. Theoretically, the expected ratio would be that most of the piglets will have erect ears, right? But observed ratios would be what happens in real life. So in real life, because of things like what you learned previously, things like your variation, right? Where things like your genetic variation, for example, crossing over and random assortment in meiosis, things like this will affect what the offspring will actually be. So let's say you have 12 offsprings, and so let's say you have let me think. The red eye. Let's use the red-eyed fly for example again. So let's say you have the red-eyed fly. Okay, he is one big letter R and one small letter R. This is a red-eyed fly, and then you have the brown-eyed fly, which is RR. Okay, so let's say they crossed over and they gave birth to 12 offsprings, for example. So 12 offsprings. Out of 12 of them, 75% of them would be brown, right? Sorry, 75%, 50% of them would be 50% of them would be brown, whereas the other 50% will be red. So obviously your expected ratio is that 6 of them will be brown, and 6 will be red. But what actually happens differs a lot. So in real life, this doesn't actually happen. Theoretically, because the sample size is so small, and you have things like your crossing over and your random assortment and meiosis, which will affect how the offspring will turn out. So in reality, it could be like, for example, 4 brown or like 8 red, or like 3 brown, 9 red, or let's say 8 red and 4 brown. Okay, it could be any of these. And this is because the sample size is so small. Okay, so the rule of thumb is that the larger the number of offspring, the closer the offsprings are to expected ratio. The closer the observed ratio is to expected ratio. Okay, so let's say 12 offspring, 50% brown, 50% red. Okay, so let's give the number up. Let's pump it up to 120 offsprings. The number won't be so drastic. Okay, it won't be like 40 brown, 80 red, or it won't be like 30 brown, 90 red. It will probably be closer to the expected ratio. It'll probably be like 55 brown, 65 red, or like, for example, 50 brown, 50 red, and 70 brown. It won't be like freaking like 30 red or like 90 brown, right? Because that's deviating too far from the expected ratio, because the sample size is really so big. So obviously the observed ratio will be closer to the expected ratio. Okay, so there's one more concept you guys have to learn: expected versus observed. So this question is testing your concept of expected versus observed ratios. Okay, 31, right? So yeah. So the question was 1,000. They said produce 1,000 piglets. Then obviously D will be wrong, right? Because a sample size of 1,000 will give you quite an accurate representation. So yeah. Any other questions? When we're answering the question, what else do we state other than small sample size? When we're answering the question, what else do we state other than the small sample size? What do you mean by that? I'm sorry, I don't really understand the question. When they ask why it is different from expected ratio, Oh, again, I told you the reason for expected versus observed, right? The reason for the deviation is things like your, I mean, we've already learned this before in previous chapters, genetic variation during sexual reproduction. So remember in meiosis, there's this thing called crossing over, where the moms and the dads homologous chromosomes will cross over and you'll exchange genetic information. And then you're thinking random assortment in when you're splitting the cells in meiosis also, splitting into gametes, you have random assortment as well. So these two things will cause the observed ratio to change also. Okay. Any other questions? If not, yeah, if you guys found this helpful, I'll flash this QR code. Sorry, let me share my screen. Give me a while. Yeah, if you guys find this helpful, you can flash this QR code and you can learn more about our weekly classes lah. So my bio is every Thursday and the timing is like not fixed. Like I can accommodate your times as well. So if you guys found this crash course helpful, just scan this QR code to learn more about what I do and to know more about how the schedule is like that and the pricing structure. So the classes will be $60 for every 2 hours. It's pretty affordable because again, I said we are purely online, so we don't need to pay rent. So that's why it's a lot easier for us to charge a lower price for students. Yeah. So any other questions so far? Oh, your earlier questions. Okay, Saga, maybe you can text me personally on WhatsApp and I can answer you. Because I don't want to hold the rest back also. Yeah. You have my number. I think I sent the link to the chat. So you can just PM me personally. Yeah. I see. Yeah. Okay, yeah. So okay, okay, I'll send the set answer to the chat. Is that okay? The crash course chat. Okay, sure. Yeah, so any other questions besides the two that Saga asked? And I'll send the answer key also lah. So if you guys are interested, just scan this QR code. If not, I will leave the call right now. But you guys can leave as well. Okay. See you guys. Why are you covering your weekly class right now? I'm just revisiting old chapters and we're just doing practice questions. So like my classes are like quite tailored to what the students actually need. So I'll live to the students and ask them what their week at. And from there, we'll just cover what they need for the week law. So it's not really a fixed kind of schedule. Yeah. Okay. Yep. See you guys. Bye bye.